can usually do it online. And then when you have an exam, if you look at the old exams, there's usually, if you need to calculate a natural log or a log-based 10, then there's usually, I give you like a list, log of this, log of this. So then you can use that, take it from that chart.
But for this one, you prefer we do it with the calculator?
Yeah. So if it's on the homework, and I don't give you that list, then go ahead and calculate it. But calculate it all the way through so you get the numbers. Yeah.
Thank you.
Okay. You're welcome.
Oh, it's time.
Okay, good afternoon, everyone. Let's get started. Okay, before we get back into the topics, let's say people had a couple questions. One, I don't know when the exams will be done graded because we have a especially large class this year. I'm not sure exactly when they'll be back, but we're getting it. We're doing them as fast as they can be. Okay, and so somebody asked me about calculating natural logs or logs-based 10. If you have need of something like that on the exams, remember we don't allow calculators or smartwatches or smartphones or any of that sort of thing. I tend to give you a chart listing some, log of this is this or whatever, and you can select from that chart. So I would expect you to actually write down the formula, plug in the numbers, and actually come up with a numerical answer at the end of a question. So again, you might want to refresh your memory of multiplication, addition, subtraction, division. But for the natural logs or the log-based 10, I would give you some information if you need that. And I guess somebody asked last time about the strike that there might be next week. I'm still waiting for more guidance from our department chair, but I think that what would probably happen with a class of this size is we can't really change the schedule for things like the next exam, so we're not going to skip a lecture, we're not going to try and reschedule a lecture, but remember that we do record the lectures, so if for whatever reason during the semester you miss a lecture, they should be available on Blackboard. Okay. Let's see. I think that was everything then. Were there any other questions about policy or anything? Anyway, okay. Okay. That was one more thing. So I think I've gotten all the regrades. And for the first exam, if you turn in a regrade, I only had 3 of them. So 2 people, the numbers went up a little bit, 1 it did not. So you can look on Blackboard and see if your number changed. But I think that was all the regrades I had. So anyway, okay, so the regrading should be done then. Last time we started talking about metabolism. So this is again that sort of new module, new section of the book. And I wanted to remind you, in case you missed it last time, that there's a little bit of information, some concepts, some definitions before Chapter 13. So you definitely want to read through that section as well as of Chapter 13. And I have some of the definitions I typed out right in the PowerPoint that you can use as your study guide again. So we went through some of this last time. Okay, and then we started with bioenergetics, and we talked about delta G, K equilibrium, and all that, and some of the equations, mass action, and all that. So definitely go through that. And we talked about a little bit about ATP. And other than ATP, like, you should definitely know the structures of ATP by now, because you went through it in the previous module. And a couple of these other structures, that's the thing I get asked like all the time, like every day in this class, is do we need to know the structures? Some of these we will be using again, so you will need to know them, like pyruvate, PEP, in pathways such as glycolysis. So yes, you should look over these structures because they hopefully will become very familiar to you. Okay, so last time we finished up some of this information and we got to the point of oxidation reduction reactions. So today we're going to talk about what are oxidation reduction reactions, how to sort of think about them and see what's going on in these reactions. This section might or might not take up the entire period, so if it doesn't, we can start moving a little bit into glycolysis, because that's a pathway you really want to get to know inside out, basically, just in all the details. So, but for now, we're going to start with oxidation reduction reactions. So the idea is that these are reactions where electrons are being moved, okay? And it might or might not be with the rest of an atom or something, but basically electrons are being moved. And this is actually where we get a lot of our free energy for surviving and for doing all these different types of work and things that we talked about last time, movement and creating energy gradient or ion gradients and all that sort of stuff. A lot of it comes down to this idea of oxidation reduction reactions in these pathways. So a lot of these different pathways we're talking about, like glycolysis and everything, we're going to have a relatively reduced compound like glucose, and it's going to be fed into some pathways. We're going to have in those pathways, some redox reactions, and eventually what happens is some of those electrons are used way farther down the set of metabolic pathways to reduce oxygen. Okay, and this is why we need to be able to breathe in oxygen. So we have glucose or other compounds like that go through some pathways. There's a number of steps, and eventually reduce oxygen. And it doesn't go directly from, hey, glucose, okay, dioxygen. We have a bunch of intermediates, electron carrier intermediates involved, a bunch of different enzymes and as well. And so there's a lot of different steps. But again, one of the key things is that this is where oxygen comes in eventually. We're going to go through glycolysis, citric acid cycle, and all that first, and then that will lead to reduction of oxygen. Okay, so the general idea is that we're going to have these organic compounds like glucose or other foods that we eat, and we're going to strip off electrons. We're going to send them through these different intermediates or cofactors and everything. It's going to be stepwise and controlled. And again, it might or might not be fast, just because something is favorable. It might not be kinetically rapid. Okay, but what exactly is an oxidation reduction reaction? So here again, I've kind of outlined a lot of your key terms and concepts. Again, we're talking about transfer of electrons. So a chemical species that loses electrons, we say that species is becoming oxidized. Another species will be gaining electrons, and we say that's been reduced. So reduced is gaining, oxidized is giving up. And so we have terms for those actual molecules. We call the one that's giving up the electrons the electron donor. Okay, it's sometimes called the reducing agent because it reduces the other molecule, or the reductant. Okay, the other molecule is the electron acceptor, so it's going to get electrons, and it's called the oxidizing agent because it oxidizes the other molecule, or it can be called the oxidant. Okay, so this can happen with just simple metal ions. Okay, this is sort of the simplest type of thing we can describe here. Here we have ferric iron, Fe3+, cuprous copper, Cu with a 1+, 1 positive charge. Put those together, we end up with iron 2+, or ferrous iron, and cupric copper. Okay, so basically, the copper gave up an electron to the cuprous copper to the ferric iron to get this. Okay, so copper gave up an electron, so it's the reductant, and it's oxidized to cupric copper. Okay, ferric iron is the oxidant, and it becomes reduced. So it's picking up an negative an electron, so it goes from 3+ to 2+. Okay. When we talk about biological molecules, so again, when we talk about doing things to glucose or other molecules like that, we're often actually going to have 2 electrons being moved. Okay, in many, many cases, it'll be 2 electrons. And if you think about it, if we're moving electrons, we're moving negative charges and everything, we kind of want to keep things, evened out a little bit. So a lot of times a proton will come along. And we actually have many families of proteins, often grouped under the title of dehydrogenases, where we can transfer a proton and a hydride. Okay, so that's 2 negative charges and a positive charge. Okay, a proton and a hydride. So again, these oxidation reduction reactions can happen in a number of ways. So that when we talk about just those metal ions, that example with just the metal ions, we're just moving 1 electron at a time. Or we can move a hydrogen atom, so a proton and an electron. So here's some compound. We're moving some protons and some electrons. Okay? Or the hydride ion, again, there's 2 electrons and 1 proton. Or something that we see very, very, very often, again, in biology, is that combination with dioxygen. So we have something with a methyl group, for example. We're going to have dioxygen, but we're actually using part of it per compound over here, and we end up getting a hydroxyl group. Okay, so the hydrocarbon is actually the electron donor, and the oxygen atom is the acceptor. And I have a diagram later that we'll go over some of this in detail. It might make a little more sense. Now, in these biochemical pathways and on these enzyme active sites, we don't always have the right side chains or functional groups on our amino acids, our 20 standard amino acids, to do all these different types of oxidation reduction reactions that we see enzymes doing. We often have these special molecules, these coenzymes, that are going to help carry the electron. So they pick it up during one reaction, and then they give it to something else in another reaction. And some of them actually pick it up from like one enzyme active site and move somewhere else in the cell and give it off again. Okay, so they're electron carriers. And even though we have thousands of enzymes, we actually have a relatively small number of coenzymes that we use over and over and over and over again. These aren't necessarily all of them, but we see these molecules NADPH and NADH, FMN and FAD. And there are also molecules called quinones. We won't go into as much detail here. We'll mention at least one enzyme that has an iron-sulfur cluster. We don't really go into too much into cytochromes in this class, but we definitely, in this part, we will focus a bit on this, and again, we will mention at least one protein with an iron-sulfur cluster. Okay, so these different coenzymes, they help transfer an electron. That means they can be reduced and they can be oxidized. They can pick it up and they can give it off again. And even though many, many different enzymes use some of these same compounds, the reduction potential or their likelihood or their ability to pick up an electron can vary because they're sitting in that enzyme active site where you have all those different functional groups and everything. So the enzyme will actually affect how easily those different molecules will pick up or give off an electron. Okay, so really, really, really common molecule. It's actually, I guess, two molecules, NAD and NADP. More than 200 enzymes, and now as we've been doing all these, genome sequences of many and metabolomes and everything, and that is millions and millions and millions of genes now. This number is, of course, much greater, but if we think about like humans and other mammals and bacteria and everything, we have over 200 known that use NAD and NADP. These are molecules. This is the basic shape down here. They're water soluble, so they can float around solution, and they move between enzymes. So they can pick up an electron in one enzyme, move somewhere else, and give it off. Okay, or give one off somewhere and go around and find somewhere to pick up a new one. Okay. So do I have a bigger picture of it? Slightly bigger on this side. Well, okay. So what is it? Okay, nicotinamide adenine dinucleotide, or nicotinamide adenine dinucleotide phosphate. So this is the basic shape down here. Okay, so this is called a nicotinamide ring up here. Down here, this should hopefully look familiar. Okay, you have adenine, ribose, phosphoryl group. Okay, so that's basically AMP right there. So hopefully you know that structure already. Up here we have another phosphoryl group. We have another ribose. Okay, so again, hopefully you know those already. So basically the new part is this nicotinamide ring. Okay, so we have this, and I'm saying there's two molecules. Okay, there's two types. The difference is, way down here, this little arrow, there's a phosphoryl group on there. Okay, so NADP has a phosphoryl group, NAD does not. Okay, might not seem that important when you have a molecule this size, whether or not there's a phosphoryl group, but to an enzyme, remember enzymes have those active site pockets. They're very, very, very specific. They're just the right size, shape, arrangement of charges, et cetera, et cetera. A phosphoryl group can make a big difference as to whether or not something will bind or not bind in that active site. So many, many enzymes will use only NAD or NADP and not both of them. Okay, so most will use one or the other. So that's helpful for the cell because you can have a pool of coenzymes that all are carrying electrons in one set of reactions, and you can have another pool, they're all ready to pick up electrons in another type of reaction. Okay, so you can have two things that are very, very similar, but you have two pools of them in the cell. You can be building up molecules, you can be breaking down molecules in different pathways. Okay, so, and I think, yes, I wanted to add, okay, so, and this is called a phosphoanhydride bond, kind of like, remember what we see in ATP, phosphoanhydride bond? And vitamin niacin is a source of the nicotinamide moiety, okay? That means that we're not very good at making this kind of molecule right here, this nicotinamide ring. We're not very good at making it, but other species can, so we make use of that, and we eat it, okay? So if we can't make it, but we can get it by eating it, and then there might be some slight modification. So when you see on your bread or something, and it says niacin, okay, that's why, because if you look at the ingredients of your bread, it's to help you stay healthy, because we need a little bit of it. You don't need a lot of it, right? It's not like you constantly need a source of carbon or glucose or something like that, right? You need a little bit of this to replace any that's lost, but these molecules, this NAD and NADP, they're reusable. They can be oxidized, they can be reduced. Oxidized can reduce. So a lot of these molecules that we use as vitamins that we need to eat, we only need a small amount just to replace any that gets lost, or if we're growing new cells and stuff like that. And so basically that's an example of what vitamins are and what they're used for. They're often these coenzymes like this. Okay. Oh, how much to take? I don't know how much. Yeah, I mean, a lot of times pills aren't like solid, niacin or something like that. There's a lot of binders or something to make it, pill size, basically. So I don't know how much of a pill is actually a vitamin when we talk about like taking a vitamin pill. That's a good question. What else is in a pill when you're taking a vitamin pill? And there's a whole other issue of when you're taking a supplement that's not regulated by FDA. Who knows what's in there, right? So, okay. So that's a good question. What are you taking when you're taking something off the shelf that says it's a vitamin? Okay.
Well, we have to know the structure of NAD and NADP.
Yep.
And so that entire structure is NAD. But
This is NAD.
But if you change that one, just that one, to a phosphate group, then it becomes NADP.
Right. So this hydroxyl group will be this oxygen will attach to a phosphoryl group, so you'll have a phosphoryl group on this instead of this hydroxyl group. Okay. So get to know the whole molecules. Again, the new part is up here. And over here, this is what happens when we talk about this oxidation and reduction. So here is the oxidized form. Okay. Notice you have a ring structure here. You have some double bonds and a positive charge on the nitrogen. Here comes some electrons. You actually have 2 electrons and 2 protons that are added. And notice you have some shifts of electrons around that ring, the addition of a proton. Okay, so this is a reduced form. And then this figure from your book, also it shows this over here. Okay, it's you can have an A side or B side. This is slightly confusing, but this is just for like people who are really into enzymes and exactly how they work. Some enzymes will basically add on the new proton on one side, and some will add it on the other side. So that's all they're saying about here on A side versus B side. Now, when we draw structures on the exam or something, I realize that you have 2-dimensional paper. And you probably don't have nice blue and pink pens. So basically, if you kind of just draw it in 2 dimensions, it kind of looks the same, okay? So basically, when the substrate is oxidized, you have this reversible reduction of this nicotinamide ring. This is the ring right here. And you get the hydride ion, H- added on. And then you either get what we call NADH, if it was NAD, or if this was NADP, you get NADPH. Okay? Okay, so these enzymes with these cofactors have this very nice thing in that if you put them in a spectrophotometer and dial the wavelength just right, you can see they absorb light. Okay, so the reduced form of NAD, so the NADH, or also the NAD pH, is going to have an absorbance peak around 340 that is not found in the oxidized form. Okay, so if you put a tube of NAD in the spectrophotometer and dial through here, you get this little, you get the red line, you get the red. So if it's just NAD plus, red line, okay. So you get some peaks over here and everything, but not down here. So basically you're dialing the wavelength and measuring at each point there. If you had the reduced NADH or NADPH, you get the blue line. Okay, so you get a peak here. Okay, so that's not just a fun fact to share at parties. This is actually really, really super important for studying these enzymes. Because look, if you dial your spectrophotometer to 340 and you start the reaction of your enzyme that has one of these cofactors in it, you can watch as the absorbance changes from, say, no absorbance to an increase in absorbance. So you can watch how quickly this cofactor is being reduced, and that tells you how quickly the enzyme is working. So you can study the kinetics of the enzyme by following what's going on with the cofactor. And fortunately, these particular cofactors, this is relatively easy to put it in a test tube and put it in a spectrophotometer, dial to 340, and just watch the, it's sort of a color change, but it's this is more UV light, so you can't really see it too well by eye. Okay, so you can study the kinetics of these different dehydrogenases. Okay. Now, another type of coenzyme, not as common in the enzymes that we're going to be discussing, but we will see it in some of the pathways, are the flavonucleotides. And these come from another nutrient. Again, if you read your package of bread and it's been supplemented, or flour or something like that, you might see riboflavin or vitamin B2. So we have FMN, which is the flavin mononucleotide, and FAD, which is the flavin adenine dinucleotide. And again, down here we have one structure that kind of works for both of them. So here is, down here again, this is our adenine, ribose, phosphoryl group, phosphoanhydride bond to another phosphoryl group. Okay, so we've seen this before. And then we have this sort of stuff up here. Okay, this is our isoaloxazine ring up here. So again, we have another ring structure, and it's attached like this. So this is shorter part up to and including the first phosphoryl group is our FMN. The larger structure that includes the adenine and the ribose and the other phosphoryl group is the FAD. Okay. So these tend to stay with the enzyme. Even though they're not covalently attached, they tend to be kind of firmly held there. So we do consider them a prosthetic group, and we call those enzymes flavoproteins. And what happens in this ring structure? Again, this can accept electrons with accompanying protons. So we have this structure. It gets reduced, and it gets reduced again. So in this case, we actually can have multiple reduced forms. We can have one's called a semiquinone or the fully reduced molecule. So we can have an FAD dot or FMNH dot like this, or we can add in another electron or proton, have the FADH2 or FMNH2. Okay? Again, we have, with these nice ring structures, we see some nice absorbance under the spectrophotometer. This is actually more invisible light here. So the oxidized flavoproteins have absorption near 570 nanometers, reduced to 450. Again, we can make use of this in the lab in enzyme assays. So we can follow the chemical reaction occurring with our enzyme, even if we have substrates and products like sugars that we can't really see too well, right? A glucose kind of looks like a fructose, clear, colorless liquid or whatever. But by making use of, the absorbance changes, from FAD or FMN, or again NAD, NADP, things like that, we can follow the reaction. Okay. so we're talking about these different things, oxidation, reduction, electrons moving around, this, that. How do you tell if a biological molecule like a glucose or a pyruvate or whatever, what is oxidized, what is reduced? Okay, like if I just give you these different molecules here, what's oxidized, what's reduced? Okay, hard to say. So I think this chart actually helps out quite a bit. When I was first looking at it, I'm like, oh, this helps make it like much clearer. So hopefully this will help you as well. So we have a lot of compounds here with carbons and hydrogens and oxygens, and I don't think we have any nitrogens here, but if you kind of picture this as, here I've got a carbon, and the carbon is sharing electrons with another atom, right? Because it's a bond, it's a covalent bond. But it kind of depends what the neighbor is as to whether or not carbon owns the electron. So this idea of owning it. So some atom's going to be more electronegative. It's going to own the electrons in the bond. So oxygen of these different atoms is going to be the most electronegative. And then it's a little more than nitrogen, which is more than sulfur, which is more than carbon, which is more than hydrogen. So that means if you have a carbon-hydrogen bond, those two electrons, we say carbon is just going to own them, okay? So here you have carbon and methane. You've got 4 bonds, so the carbon is, supposed to be sharing those electrons with the hydrogens, but we can think of it, we can kind of picture it as the carbon's kind of greedy. It's actually going to own all of them. It's actually going to own all 8 electrons. That's 2 electrons per bond. So we think of methane as that carbon is actually owning all the electrons, even though that's really in a bond being shared with the hydrogen, okay? Now we come down to ethane. Okay, we have carbon, this carbon with 3 hydrogens and then with 1 more carbon. Well, with the hydrogens, again, it's going to own all those electrons. But it has one neighbor that's another carbon. So 2 of the same type of atom, they each are going to own 1. So if you compare methane to ethane, the carbon up here is owning all 8 electrons, but down here it's only got 7. Go down a little farther with ethene, and you have a double bond, so the carbon is now sharing 2 bonds with the other carbon, so it only gets 1 electron from each of those 2 bonds, and then both electrons from each of the bonds with hydrogen, so it's down to 6 now. And then now we're going to start bringing some oxygen. So oxygen over here is more electronegative, so it's going to own them, own the electrons, even the carbon's going to own the electrons when it's bonded with hydrogen. It's going to own 1 from the other carbon, but it's not going to own any with the oxygen. So now we're down to 5. Okay, so you can kind of continue on with that. Here's a triple bond to a carbon. Here's a carbon double bond oxygen, carbon double bond oxygen with, so you have all these different combinations basically. And we get down to carbon dioxide, carbon and 2 oxygens, double bond to each of the 2 oxygens. It doesn't own any of them, okay? So we think of this functional group here with carbon. You can compare with a chart like this whether it has more electrons or fewer electrons compared to like this functional group compared to this functional group. Okay, so you can say if it has more electrons, it's more reduced. If it has less electrons, fewer electrons, it's more oxidized. Okay, so, and this is kind of another way of looking at, here's our alkane, right? That carbon is owning a lot of electrons because it has some hydrogens as bonding partners, okay? So this is, of these different, down here it's, it doesn't own any because it's with those oxygens. And then in between it might have a bond with an oxygen or 2 bonds with an oxygen and some bonds with other carbons or hydrogens or whatever. So the thing is to look at this functional group at the end and say, well, this one is most reduced because the carbon of these different carbons in these different functional groups, this one owns the most electrons. It has the most electrons. It has the most, sort of negative electrons, or whatever. And down here, this one, carbon dioxide, is the most oxidized because it doesn't really have any. Okay, so it's kind of, think of it as, sure, technically it has bonds with these oxygens, but the oxygens are, we can think of it as owning all the electrons. Okay, so that's how we would, so kind of look through this chart, think through this chart, and then bring it to something like this and say, okay, here's some molecule, and here's a functional group at the end. And think about, okay, going from an alkane to some modification that makes it an alcohol, it's been oxidized, okay, and on further and further. Okay, so again, when you have something added or subtracted to a bond, so in this case, think of this as being within a molecule. So there's something out here, something out here. Here's a single bond between two carbons, and each of them also has some hydrogens and then something else attached to out here. When this undergoes a reaction that, there should still be one hydrogen on each of these. Okay, I'll have to fix that. But each of them has lost one hydrogen and gained an additional bond to the other carbon. So the carbon has undergone oxidation here. And if you look at this and this and this, that also gives you this idea. See, the oxidation often occurs with dehydrogenation. Right, so when we're talking about reducing cofactors, we're saying the electron comes and a proton, maybe a hydride is transferred and everything transferred. It's also the opposite when we talk about oxidation. The electrons might go away. Often you're also losing some hydrogens along with that, some protons. Okay, so let's look at an example in a molecule that we'll see in some of these pathways. So we have a lactate molecule. Here it is, now it's in the middle of this molecule, the carbon is, lactate, this carbon, oxygen bond. And then we have pyruvate over here after this reaction. So what happens here? We go from lactate to pyruvate. We lose two protons, this one, and two electrons to get the carbon-oxygen double bond. Okay, so the lactate is oxidized to pyruvate. Okay, I'm looking at this. Because it's losing the electrons, okay, and we go from pyruvate to lactate, we're adding electrons. Along with those electrons comes two protons. So we're adding electrons, and you get lactate. Okay, so that's looking at the other way around. So you can go to either direction. Okay, so that's all I'm going to say about oxidation reduction. So hopefully those different diagrams and thinking of it through those different ways will help you identify what's going on in a reaction. Has something been oxidized or reduced? Okay, and there's some questions that might be in the homework of the old exams, but there should also be some extra practice questions on a page on Blackboard as well. It was under the table, or I'm sorry, it was under Module 1, and I copied also into Module 3. So there's some questions there for Chapter 13 related topics. Okay, so since we have lots of time, I'm also going to bring up, if I can find it, Chapter 14 lecture. So we'll start a little bit of the glycolysis. Okay, so glycolysis. Okay, glycolysis is a pathway that's super important. It's a very central pathway in metabolism and everything. It's also a great example of many, many different features of biochemical pathways. So, almost any biochemistry course is going to go through glycolysis, or MCATs and all those other things. I'll ask you something about glycolysis. So what is it? So this is named from the Greek. So sweet, loosening, You see a lot of things with lysis in biology, breaking up things. You always think of it as breaking up things. Okay, so we're going to take glucose, one type of sugar, and break it down. It's going to be in a number of steps. You want to get to know all the steps, okay? I tell you that right away because it takes some people, they want to have a little extra time to get to know their pathways before the exam. So definitely get to know the glycolysis pathway in detail. There are specific steps, enzyme catalyzed reaction. In the end, we take this 6-carbon sugar, we're going to end up with 2 molecules of pyruvate. We're going to end up making some ATP from ADP, great source of energy, right? And we're going to reduce some NAD to NADH. So some of these cofactors we were just talking about, immediately NAD becomes important. It's going to help us collect some of this energy that can be used later actually to make more ATP. Okay, and the pyruvate, depending on the species and everything, it can be aerobically oxidized by a lot of species, like by us, okay? But it can also be used for a lot of different things. You can make amino acids and other things from it. So this is an example. We talked last time about pathways. You can have divergent and convergent pathways. You can make pyruvate from glycolysis. You can actually make it from other things. You can break it down further to get more energy, or you can make a lot of other different types of things from it. So a lot of these different pathways, even though we kind of focus on here's glycolysis, here's citric acid cycle, et cetera, there are actually a lot of different connections to many other pathways in the cell. Okay, so as we break down our glycolysis, we get our pyruvate. That might go on for other things, but we make some ATP, we make some NADH. Yay, we have a source of energy. Okay. Now glycolysis, again, we can make pyruvate, whatever. So down over here, it's really one of the pathways for glucose. Okay, so oxidation via glycolysis, we get our pyruvate. It's great. We can get some energy. If we're species like us that can actually take that pyruvate and continue breaking it down further, theoretically, one could get a huge amount of energy from that glucose. I mean, in practice, we don't actually get quite that much, but if we broke it down all the way, glucose to pyruvate, and then continue on to carbon dioxide and water, and use all those reduced cofactors to make ATP and everything, you can actually get quite a bit of energy from glucose. But that's not the only use of glucose. We don't always want to be breaking it down, right? I mean, sometimes we're eating our lunch, but there's a lot of times we're not eating our lunch, so sometimes we want to keep some of that glucose for later times. So we might want to store it. Since as people, we tend to store it as glycogen. If we were plants, we might store it more as starch, okay? Plants also make sucrose. There are other ways of storing it as well. We also may want to build things with it. So extracellular matrix, cell wall polysaccharides. A lot of things are actually made out of glucose, or glucose is used to make other sugars that are then used, like the carbohydrates on proteins that are on our cell surface. So it's used for a lot of structural polymers, it says here. And it's also a source of 5-carbon sugar. So glucose is 6-carbon sugar. And we'll mention briefly this pentose phosphate pathway later. So we can do a lot of things for glucose, or with glucose, I mean. So all these different pathways, again, they're going to be regulated. We're going to focus a lot on glycolysis, but that's not the only thing we do with glucose. And we're going to have ways of sending glucose to whatever pathway is going to benefit the cell at that time and under whatever circumstances. Okay, so let's talk a little bit about why we care about glycolysis. Okay, what's important about glycolysis? What are some interesting things about glycolysis? Okay, so this is found in almost all species. Almost everything on Earth has glycolysis. Okay, there are a few that don't, like Rickettsia, the organism that Causes Lyme disease. Anyone heard of Lyme disease? You get bitten by a tick on the East Coast there. I don't know if you have it so much out here. Do you get, no? Okay. Yeah, it's out here too? Yeah. So you want to be careful about ticks because you can get Lyme disease. You can get from the Rickettsia bacteria. So Rickettsia is one of those species that actually, it's an obligate intracellular parasite. So it can't actually live on its own. It has to live in something like ticks or in us or something like that. And so it doesn't actually have the complete pathway of glycolysis. It can get by getting certain molecules from its host. But almost everything else on Earth does have it, other than things like, let's say, that actually live inside of a host cell. Okay. Glycolysis is also, for some organisms, the sole source of metabolic energy. So remember I said you can make a little ATP, a little bit of ATP, and that's what some organisms, that's basically all the ATP they can make. Okay, we can do better. We have other pathways that we can continue breaking down pyruvate and everything. It's also a pathway with the most carbon flux in most cells. Okay, what's flux? That means that when you have carbon-containing molecules, a lot of them are going to go through this pathway. There's lots of pathways, but this pathway. Think of it as like if you have lots of little pipes in your plumbing, there's one big pipe in your apartment building that's going to have the most flow. This is the pathway with the most flow in the cell. Okay? It's also a good example. So, this is one of our reasons for teaching things. Is not just we don't want you to just memorize things for the sake of memorizing things. We want you to actually learn about different concepts. So this is important pathway, regulation, different use of cofactors, et cetera, et cetera. So this is a really good example for all that. This is also the first pathway to be determined. So some of these enzymes in this pathway have been studied since like the late 1800s when they first broke open yeast cells and said, hey, some of this chemistry is still happening even though we've killed the yeast cell. The yeast cell's dead, but something's still happening chemically to sugars, okay? So people figured out that, hey, there's these different enzymes, and this does this, and then this does this, and this does this. And so it took them about 40 years, but they, don't worry, they got their PhDs long before that, but after about 40 years, they figured out that there was actually a pathway to follow. Okay. And in studying this pathway and a few related ones, people figured out how do we actually purify these proteins when we had to memorize that stuff about different columns and stuff and different concepts. How do you purify protein? Well, this was something that was kind of worked out back in the 40s before they could go online and buy whatever columns they wanted to buy. They actually had to devise ways of doing this, come up with some concept about how do you separate this protein away from all these other proteins, right? So then they had to make whatever materials were needed for that. So things took a lot longer back then. They figured out why, what is NAD for, what is ATP for, they got them in the cell, what are they doing? And also phosphorylated compounds. Okay, I've talked before about phosphorylated amino acids and phosphorylated sugars and stuff like that. This is another very important use of phosphorylation. Okay. Now, so we talk about glycolysis, and I mentioned kind of back here this idea about, breakdown, oxidation, use of oxygen, all that other stuff. The actual steps of glycolysis, these 10 steps, it's actually anaerobic. Oxygen doesn't play a part until another pathway farther down, okay? So this is what we actually call a fermentation. A fermentation, like when you ferment foods, okay? This is the idea. You have an anaerobic, no oxygen needed, breakdown of organic nutrients, in this case glucose, but other fermentations, if you're making sauerkraut or yogurt and things like that, you might have a fermentation if it's an anaerobic breakdown of organic nutrients to obtain energy. Okay? Why is it so central? Why is it found everywhere? Why is there so much flux going through it? Why, Well, way back, okay, not a few weeks or a few months, but we're talking about probably about 3 billion years. This was probably a key pathway that was helping to produce energy. And it worked. So if it worked, you keep it, okay? It's so old that it's before green plants. So you didn't have photosynthesis, you didn't have free day oxygen out in the atmosphere to any significant extent. So you got anaerobic conditions. You need a pathway that works under anaerobic conditions. So this kind of came together. We want to extract some free energy, we want some energy, we don't have any oxygen around, what are we going to do? Okay? So this is a way of breaking down molecules. I'm sorry, I'm hearing some talking because people hold their conversation till later, please. I know you think you're whispering, but believe me, we can all hear you. Okay. So how to extract some free energy from some sort of nutrient or food or something, but you don't have oxygen around. Okay. So what happens in glycolysis? We're going to get that first molecule activated, okay? We're going to do something to it. We're going to add some phosphoryl groups to it. And then we're going to have some more steps that we're going to start getting back some of the energy from it. And there's still people talking like way in the back, but I'm sorry, this place really echoes, so please hold your conversation till after class. Or do we want a 2-minute break? Okay, I'll give you a 2-minute break. How's that? Because we still have like 20 minutes. Okay, 2-minute break. I'll have a little beverage. And if you need a topic of conversation, you can think about why do we drive on parkways and park on driveways. Okay, there you go. Okay, are we ready? Okay, let's continue. Okay, so here's glycolysis. Ta-da! Okay. So, looks super complicated. There's 10 steps, but believe me, almost everyone does really well in learning all these steps. And this diagram is giving you lots of different pieces of information all together. You have the names of the molecules and when ATP is made or used or whatever, and other molecules. You have the actual structures over here. One thing I don't like that the book does, so don't do this in the exam, is if you have a phosphoryl group, show me what an actual phosphoryl group looks like. Don't do this little thing where they draw a P with a circle around it. Okay, show what an actual phosphoryl group looks like if you're drawing a phosphoryl group. But basically we have 10 enzymes. Here are the names of the enzymes. Okay, here are the compounds starting up here with glucose. Oh, there's our pyruvate down at the bottom that we were talking about, and here's some ATP and NAD and everything. And you'll notice there's 2 boxes. We have 5 steps up here. We call this the preparatory phase. And we have another box down here called the payoff phase. And that's to sort of help us think about what's going on overall here. In this preparatory phase, we're going to take our glucose. We're going to use some ATP to phosphorylate it and then phosphorylate another molecule farther down the pathway. And, well, that might not sound too good. I was just saying how, like, we've got all these organisms using glycolysis to get energy. Why would we burn up some ATP in glycolysis? Well, we need to get it going. We need to get this set of reactions going. We're going to invest some ATP in this preparatory phase. Down here, oh look, we get some of our ATP back. Okay? We use 1,2, we make 1,2. That doesn't sound very good. But wait a minute, if you look carefully, and we'll go through these steps in some detail, you're starting with a 6-carbon compound, and then there's a big step of chopping. You chop the pink half from the blue half, you get 2 compounds. Each of these goes to the next half, so you get 2 ATPs here, you get 2 ATPs there. You're going to get more ATP out than you put in, so you get a payoff, okay? So that's maybe a good investment. You also get some reduced NAD, so you get NADH. If you're an oxygen-breathing organism like me, okay, then you can use this NADH later to make more ATP, okay? Certain organisms that, again, have glycolysis as their only ATP-making pathway, or maybe living anaerobically, some organisms cannot take this and then use that to make more ATP, okay? So different organisms, different species can do things differently, okay? Okay, so let's talk about some of these pathways, or some of these enzymes in some detail. So we're going to start going through some glycolysis today. That gives you a little extra time to start learning the different molecules, what's going on in the reaction, how to kind of connect what's going on with the names of the enzymes so you can kind of remember them a little bit and help you prepare a little more before the exam comes up. Okay, so we're going to start out with something called hexokinase. Okay, this is one of our steps. We're going to prepare our glucose for some of our later steps. And hexo, okay, it's a hexose. It's a 6-carbon sugar. That's a hexose. And it's a kinase. The enzyme is a kinase. So what's a kinase? Basically, a kinase takes some molecule. Okay, we talked about, right, protein kinase. This is the sugar kinase, a hexokinase. We're going to add a phosphoryl group. We're going to get that from ATP. So here we have glucose, goes to glucose 6-phosphate, phosphoryl group from ATP, and here's in pink here is, or red is the numbering. 1, 2, 3, 4, 5, 6. So it's the phosphoryl group replacing the hydroxyl group on the 6 carbon. So you get glucose 6-phosphate from hexokinase. Other kinases might do it somewhere else. Hexokinase, carbon 6, whether it's glucose or mannose or some other certain other sugars as well. Okay. When we talk about this ATP being used, notice down here there's this little magnesium 2+. Okay, sometimes you'll see this added in drawings of different reactions like this or whatever. This magnesium, it's not added onto the molecule in any way. This is often needed in an active site to help with the interaction of the ATP. Remember, the ATP has 3 phosphoryl groups that has a number of negative charges. Magnesium 2+, right? Divalent cation, positive charges. It helps in the binding of ATP to a lot of different molecules. Okay. So again, we're using up some energy. We're going to use some energy in this step. Okay. Now we have our phosphorylated glucose. We need to still do some more kind of changing things around, adjusting things before we can really start getting energy out. So we're going to take our glucose 6-phosphate, convert it to fructose 6-phosphate. This is an aldose. This is a ketose. Okay, these are the ring forms, but if you draw the open chain form, you'd see that better. What's going on? We have phosphohexose isomerase. I don't see any additions of carbons. I don't see any removals of carbons. That phosphoryl group's just sitting there. Okay? We have some shifting of bonds but we haven't gained or lost any atoms, okay? That's what an isomerase does. It moves things around, okay, but we don't add or subtract like a phosphoryl group or carbons or anything like that, okay? And, well, not going into the details of this reaction. Yes, question?
I have a question with the preparatory phase with the hexokinase. I noticed that the 6 carbon right there, it's on the right side. If you go back to where the reaction is, it's on the left side. Does it matter? If you go back to the whole chart, it's on the left side.
Okay, so if you draw a molecule, this way of drawing a molecule, we're not indicating anything really. Gosh, how should I say it? Well, this is the same, right? The hydroxyl group's just basically on the other side, which is really the same molecule. Right? I mean, it's not, you have rotations around the single bonds. So you can draw it on either side, it's still the same molecule. Okay? Yeah. Okay, what was I talking about? I was talking about phosphohexose isomerase or phosphoglucose isomerase. We won't go into details of this mechanism, but I could tell you an entire seminar on this because I saw the structure for this during my postdoc. But yes, question? It depends on the isomerase. There might be different mechanisms for how you go from substrate to product. There's a lot of different enzymes that we would call an isomerase. So the mechanism might be a little different, but basically what we're having is that you're converting from one molecule to another, and you're not like adding a whole functional group or removing one, okay? So these, again, same number of carbons, same number of phosphoryl group, same, and everything. Same number of oxygens, okay? Okay, so again, in this class we don't necessarily go through so many details of the mechanisms, maybe 1 or 2 enzymes if I put them on the slides, okay? Okay, so we're still not quite ready to get energy out of our 6-carbon sugar. We still need to invest a little bit more. We're going to use another ATP. So we go from fructose 6-phosphate, use ATP. Oh, there's a little magnesium there again. Okay, ATP, use ATP. We're going to now phosphorylate on the 1 position. Okay, so we have fructose 1,6-bisphosphate. Okay, bis means 2, but it means 2 that aren't connected to each other, okay? So bisphosphate means we got a phosphoryl group over here, and we got a phosphoryl group over there. The 2 phosphoryl groups aren't covalently attached to each other. They're attached to different parts of this fructose, okay? So fructose 6-phosphate to fructose 1,6-bisphosphate. Let's think about what might do that. Phosphofructokinase, okay? So let's think about that. This is a phosphofructo, okay? And the enzyme a kinase uses an ATP to donate a phosphoryl group to a molecule. So again, the name goes with what it's doing. Okay, so if you're trying to remember what are the molecules, what are the reactions, what are the enzyme names, remember what a kinase does, remember what a phosphoryl group looks like, what a fructose looks like, things like that. And then this is phosphofructokinase 1. There can be a phosphofructokinase 2 in other pathways and everything. Okay. Now, one thing that's very important here about this particular use of ATP in glycolysis, this is the 3rd step, right? It's not the 1st step, but this is a very important committed step. This is an irreversible step. This is going to go forward And it's at this step we're actually going to find later, as we talk about things like regulation. This is the point where you get this fructose 1,6-bisphosphate. This can continue down through glycolysis. If you have glucose or glucose 6-phosphate or fructose 6-phosphate, those three molecules can all be used for many different things, okay? Going back to this idea of you can make glycogen, or you can make carbohydrate glycans on proteins and lots of other things. Once you get through this step where you're using ATP, you're investing in this ATP, and you get to here, then you're going to continue down through glycolysis. Okay. If you have a lot of ATP around in the cell already, okay, the cell's got a lot of energy, you don't need to be going through glycolysis, you don't need to be breaking down your glucose, okay? Well, this ATP can come and actually bind to the enzyme in a way that you lower the activity of the enzyme. You lower the activity through this pathway, leaving fructose 6-phosphate and those other molecules to go be used for something else. Okay, so you don't want to use up all your glucose as soon as you make it, right? You want to store some, you might want to use it for other things. If you have lots of ATP around, you don't need to go through glycolysis, you don't need to use up all your ATP. Okay, but once we get to fructose 1,6-bisphosphate, now we can really start doing things to our molecule. Okay, so we're going to chop it in half. Okay, we're going to take 6 carbon fructose 1,6-bisphosphate, so 6 carbons, 2 phosphoryl groups, chop with aldolase, and you get dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And now if you look at this, now we have some little numbers here in parentheses. This is showing you where these parts come from. So we chop in the middle, okay, and you have, this was 1, this was 2, this was 3, this was 4, this was 5, this was 6. Okay, that's why you have the numbers in parentheses. That tells you where they came from. That can be important with certain types of questions or even sort of the understanding of the pathway. Okay. Once we have these 2 different molecules, oops, back up here, 2 different molecules, really one of them is going to continue down the pathway. So we're going to convert one of these into the other one. And they're both 3 carbon sugars with a phosphoryl group. Again, we have a ketose, right, and an aldose. We're going to use another isomerase. We're going to take this dihydroxyacetone phosphate and make another glyceraldehyde 3-phosphate. This is an isomerase. Remember we saw that isomerase just a few steps ago. This is another one. This is also this can go back and forth between a ketose and an aldose, okay? In fact, it has a very similar mechanism. There's a lot of things right in the active site they share, okay? Very similar mechanism. But in this case, we have 3 carbons, so it's a triose, 3 carbons, phosphate, right? Got a phosphoryl group there, isomerase. Okay, so you're going from ketose, aldose, dihydroxyacetone phosphate, glyceraldehyde 3-phosphate. So this glyceraldehyde 3-phosphate and this one that was made directly from fructose 1,6-bisphosphate by aldolase, they go through the second half, the payoff phase. So you have 2 copies of glyceraldehyde 3-phosphate going through the second half of glycolysis. Okay, so we want to pause here and think about what happened to those different carbons, starting with glucose, but in this diagram we've already converted. So we went from glucose 6-phosphate, fructose 6-phosphate, and then we added that other phosphoryl group. So here we have it drawn out, not in the ring form, but in the extended form. Here's our 2 phosphoryl groups, and we have carbon 1, 2, 3, 4, 5, 6 here. And aldolase, again, is going to go chop right down the middle, okay? and you get your dihydroxyacetone phosphate, you get your glyceraldehyde 3-phosphate. The triose phosphate isomerase can convert them one to the other. Okay. So what people can actually do is say, what if carbon's usually carbon-12, most common isotope of carbon, but we can go buy this molecule fructose 1,6-bisphosphate where we have one of the carbons, let's say carbon 1, is a carbon-14 instead of a carbon-12. Okay, so now if we put that in a test tube and add aldolase and triose phosphate isomerase, we can say that carbon 1, which is a carbon-14 isotope, we can say, oh look, it ended up over here in dihydroxyacetone phosphate, and then we use triose phosphate isomerase, and this is kind of flipped over, but this will end up over here, okay? So if you kind of look at this and you kind of flip the blue one over, you can see that, or I guess flip the red one over because they have the phosphoryl groups down at the bottom. So carbon 1 ends up over here. If you did it to carbon 2, so everything is carbon 12, but carbon 2 is actually a carbon 14 isotope, follow that, it's going to end up over here. And then you're going to, bless you, from that you're going to make glyceraldehyde 3-phosphate. So carbon 2 ends up here in the middle of glyceraldehyde 3-phosphate. You can do that with each one. Carbon 3 ends up down here, which ends up here when we flip it over. Okay, carbon 4 ends up over here or here. 5 is in the middle of the second half, so it's there and there. 6 is down by the other phosphoryl group, so it's there and there. So 1 and 6 are both by phosphoryl group, so they both end up here, right? 2 and 5 are both in the middle of their half of the hexose. They end up in the middle. And then 3 and 4 are near each other where it's chop, and you get them here. Okay, so people can actually follow chemical reactions by using these different isotopes of certain atoms. You can get these things made, or you can make them. Nowadays we would probably just buy them, I guess. But okay, so they get made, and you can follow where each atom ends up after a step or a few steps down the pathway. And this is used to work out a lot of chemical mechanisms or enzyme mechanisms, as well as pathway mechanisms. And you can ask questions like, oh, is this enzyme involved here, or how does the enzyme do it, or what intermediates are there, and things like that. So one of the things, if you've started looking at any of the extra problems or old exams, or maybe in the homework, I don't remember exactly which questions I gave you this semester, but you might be asked to follow a carbon-14 labeled molecule through some pathway, whether it's glycolysis or citric acid cycle or both. And this is how you do it. You think about where it is in one molecule, you think of what the enzyme does to it, and you can follow that carbon goes and becomes a specific carbon in the product, and then that becomes a specific carbon in the next product. And that's how you do it. And this diagram, if you're not quite sure, if you're trying to do some of the problems, this is one of the key problems, because this is where that big aldolase chopping happens, and then you've got the 2, and does it go up or down or whatever. So this diagram is very helpful for learning that. But you can also follow oxygen. Okay, oxygen's usually isotope 16, but you can get oxygen 18, for example. Nitrogen, you can have 14 or 15, things like that. So you can do this with other atoms as well. A lot of our questions tend to be with carbon-14, but we could also be following the oxygen or following a nitrogen in another pathway, for example. Okay, so see you Tuesday, I guess. Today's Thursday. Okay, have a good weekend. maybe look on the web and find out some more background of that. But basically, you can have different versions